Okay, this is your Chapter 11 study guide that will walk you through your study guide so you can get ready to ace that next test. If you look at question 1, it says, if you take two animals that are different, such as a horse and a donkey, and they produce a cross between a horse and a donkey, what is that an example of? So here we see the word a cross between a horse and a donkey. Now, this would be a mule. That's not what the question is asking, though. It's asking what would the mule be an example of? Well, a mule is neither a horse or a donkey. So this would be an example of incomplete dominance because it's a cross between a horse and a donkey. Now, there's another word that is used for incomplete dominance, and that is a hybrid. And we talked about these hybrid traits when we did our genetics lab, when we flipped the coins and rolled the dice. So an example of a cross between a horse and a donkey would be a mule. An example of that would be a hybrid. All right, number 2, know how to draw and complete a Punnett square along with the terms heterozygous and homozygous. So it's best if we start with the terms. So here we have the word heterozygous. Hetero means different. And here we have one allele that is dominant and the other recessive. And dominant traits are shown by capital letters. Recessive traits are shown by lowercase letters. So here's an example. We have a dominant trait and we have a recessive trait, and that is heterozygous. Because remember, hetero means different. Well, if hetero means different, we know that homo means the same. And in homozygous, both alleles for a trait are dominant or they're both recessive. So here we would have homozygous dominant. If both the letters were lowercase, they would be homozygous recessive. Now, as far as the Punnett square goes, here we have two organisms. This is a homozygous recessive because they're both lowercase, homozygous dominant because they're both uppercase. So you put one on the side, one on the top, and then what you do is for these A's on the top, you simply bring them straight down. These A's on the side, you simply bring them straight across. So if I'm looking at this, I'm combining this capital A or this dominant A or allele with this recessive allele. So I would have 4, after I fill this Punnett square in, I'd have 4 heterozygous organisms. All right, number 3, know why the principles of probability can only predict what offspring may look like and not determine what it actually looks like. Well, in genetics, the passing of alleles is as random as flipping a coin or rolling even in odds using the dice. You do not get to choose what you pass on. It is random. Your body will make gamete cells, which are sex cells, but as to what sex cell actually gets used, or the one that will be used for sexual reproduction, you don't get to choose. It is completely random, and that's why it can only predict what your offspring may look like. It will not actually determine what your offspring looks like. All right, number 4, know what the principle of independent assortment is. Independent assortment is when different genes independently separate from one another when reproductive cells develop. I got that from nature.com. I thought it was a great simplified definition. So imagine we have this right here, which is for hair. And the other one, C, that we used is for height. And we just chose that. It could be any letters. I just chose H and C. So the gametes that the H, and we have gametes, so the sex cells that the dominant H and the recessive H go in are not dependent upon where the dominant C and little c go. Hair color has no impact on your height whatsoever. All right, hair color is a different gene than the gene for height. They separate from one another when the gametes form. That is independent assortment. We talked about that the other day in class. All right, moving along, number 5, variation in human skin color. Is that controlled by multiple alleles or is that controlled by multiple genes? And what in the world is the difference? Well, variation in human skin color is known as polygenic traits, which means many or multiple genes. Now, multiple genes are many genes that affect one trait. So in this example, human skin color is one trait controlled by many genes. So there are multiple genes that affect skin color. That's an example of multiple genes. Well, multiple alleles is when there are multiple variations of a single gene, and this is known as an allele. So for example, we have a heterozygous organism here. We have a dominant H and a recessive H, and we see 2 alleles, a recessive allele and a dominant allele, and this is for one trait. In the previous previous example, we used H for hair color. And there we have two alleles, but two alleles affecting a single trait. Here, multiple genes that affect one trait. So we have one trait here, hair color, that's affected by multiple alleles. We also did this with blood, with the A blood type, the B blood type, the AB blood type, and the O blood type the other day. Number 6, figure out what the diploid number is given a haploid number, or figure out a haploid number given a diploid number. This is probably one of the easiest questions you'll ever come across if you know what diploid and haploid means. So let's take a look at this. If the diploid number is 14, what is a haploid number? Well, haploid sounds like half and contains half the number of chromosomes. If the diploid number is 14, half of that would be 7. Now, in haploid cells, there is one set of chromosomes. There's one set. And so our single set in this example would be 7. 2 sets of the chromosomes would be 14 because diploid means 2. Well, if the haploid number is 21, what is a diploid number? Well, di means 2. We would double or multiply 21 by 2. There are 2 sets of chromosomes in a diploid cell. 21 plus 21 is 42, or you could do 21 times 2. Here's another example. What is a diploid number if the haploid number is 23? Well, if the haploid number is 23 and we're trying to get to the diploid number, we got to multiply it by 2 or add 23 plus 23, which is 46. If the diploid number is 24, what is the haploid number? What is half of 24? And that would simply be 12. Okay, number 7. What and how many cells are formed at the end of meiosis? Well, 4 haploid cells are formed, and they are all genetically different. And haploid cells are gamete cells, which we'll talk about in just a minute. So here's an example of meiosis. It's gone through meiosis 1, so prophase, metaphase, anaphase, telophase the first time. Meiosis 2 is prophase 2, metaphase 2, anaphase 2, telophase 2, and at the end, we wind up with 4 gamete cells, or 4 haploid cells that contain a single set. Here we have 2 diploid cells that contain both sets of chromosomes. Here we have haploid sets. They only contain half or one set of chromosomes. And you can see that all of these are genetically different. All right, number 8, unlike mitosis, meiosis results in the formation of what? And when that's just what we talked about. Gamete cells, which are 4 haploid cells, and they are all genetically different. So 7 and 8 kind of go together. Number 9, know that linked genes never assort independently. The genes are linked together. These genes are found on the same chromosome. So here we have some linked genes. They are found on the same chromosome. Here we have 2 genes that are not linked because they are not found on the same chromosome. They are found on different chromosomes, and the same thing here. So linked genes are going to be close together, and this will help lead into number 10. These are not linked even though they're on the same chromosome because they are far apart. So the genes that are linked are going to be found in the same chromosome close together, and this leads us into number 10. So 9 and 10 kind of go together because this will make more sense. When is crossing over more likely to happen with genes on a chromosome? When the genes are closer together or farther apart. So these genes are closer together, so they're more likely to cross over. These genes are farther apart, so they're less likely to cross over. And because these genes are closer together, they're more closely linked to one another. These genes are farther apart, so they're less closely linked. So when they are closer together is when they're going to cross over. So 9 and 10 kind of go together. Number 11, know that the F1 and F2 generations represent the first and second generation. What generation makes up the F1 and F2 generation? So we're given the fact that F1 means the first generation, F2 means the second generation. Now, where does the F1 and F2 generation come from? Well, initially they come from the P generation. So the P generation stands for the parental generation. Now, the parental generation somewhat helps make up the F2. So the parental generation would be like your grandparents. Your parents would be the F1, you would be the F2 generation. So parental generation produces offspring that makes up the F1 generation. The F1 generation produces offspring that makes up the F2 generation. So the F1 and F2 generation come from the parental generation. And one more time, the parental generation is like your grandparents. Your grandparents produced your parents. That would be the first generation. Your parents produced you, and that would be the second generation. Number 12, what do scientists mean by genotypes? What do scientists mean by phenotypes? Scientists have done us a huge favor here that makes this very easy. You just ask yourself, what word is inside the word genotypes? And that is simply gene. It is the genetic makeup. You cannot see it. And these are written as letters such as capital R, lowercase r, capital T, lowercase t, capital A, lowercase a, capital A, capital B, lowercase b. You can't see it. Just looking at the letters, you don't know what the organism looks like. Well, if the genotype is a genetic makeup, phenotype is the physical makeup. It's the physical traits you can actually see, such as blonde hair, light skin, blue eyes, brown eyes, freckles. If you can see it, that is the phenotype. The phenotype is made up by the genetic code, the genotype, and that brings us to number 13. You need to know that the phenotype and genotype make up an organism's characteristics. So genotypes and phenotypes make up the organism's characteristics, and we just talked about the difference between those two. So genotypes, which is the genetic code, codes for the phenotypes, what we see. So for example, we have this genotype here, capital B, lowercase b. That codes for the phenotype, what we can see, which is brown eyes. All right, number 14, what does a gene map show? This is an easy question as well, because a gene map is exactly that. It's a map of the genes. It shows where genes are located on chromosomes. We looked at this when we did DNA, and we talked about chromosome number 20. All these diseases are shown where they're found on chromosome number 20. All the diseases, we know where they are. They've been mapped out. And that's what we call a gene map. Number 15, how many chromosomes do gametes have? Well, we know gametes are sex cells, and because they are sex cells, we know they are haploid cells. They contain half the number of chromosomes. They have one set. This doesn't mean they have one chromosome. They have one set of chromosomes. So in humans, gametes have one set of chromosomes, and that would be 23 chromosomes. And half of 46 is 23. We have 46 chromosomes. If you're wondering how we got 23, all we did was take half of 46. All right, almost done. We only got 4 questions left. 16, why did Mendel choose to study pea plants? And there's several reasons. He could grow a lot of them. He could easily see the traits, and he didn't have to wait long periods of time for the peas to reproduce. So those are some of the few reasons he chose to study pea plants. 17, what do scientists mean by homologous chromosomes? For some reason, students always struggle with this, and it's not too difficult to understand. Homologous chromosomes are chromosomes that pair up. For example, this means chromosome 1 from dad pairs up with chromosome 1 from the mom. Here we have homo. It means the same. Chromosome 1 is going to pair up with this same chromosome from the other parent, chromosome 1. So here we have some homologous chromosomes. We see that these are pairing up, and that is because they have the same alleles, and we get one recessive allele from one allele from one parent, one allele from the other. They could both be recessive like they are here. One parent gives off a recessive trait. The other one gives off a recessive trait. This would be homozygous recessive. Here we have a dominant trait given off by one, a dominant trait given off by the other. That would be homozygous dominant, homozygous recessive, and here heterozygous. So these are the same chromosome number. They're pairing up, and that's so we can get these alleles that match up. And if we get a hybrid trait or incomplete dominance, we get some crossing over that happens. Homologous chromosomes. It just means that each chromosome number pairs up with the other chromosome number. That's it. They're chromosomes that pair up. All right, 18. How many sets, not the chromosome number, of chromosomes are in a diploid cell? And what part of the word diploid makes the answer obvious? Well, di means 2. So there are 2 sets of chromosomes in a diploid cell. Di is a diploid, which is a dead giveaway to this. So di is diploid, which means 2 sets. That's a dead giveaway. That's all you need to know. All right, how do you give the ratios of a Punnett square? You count the genotype or the phenotype of the traits you are looking for and put this out of the total amount. Here's an example. We have a dihybrid cross, and there's 2 ways. So round yellow. We know round is given off by a capital R. Yellow is given off by a capital Y. 1, 2, 3, 4. Not here because we don't have at least one capital Y. 5, not here, 6, 7, 8, and 9 right here. So that would be 9 out of 16. We could also write it like this. And if I look for round green, there would be 3. Wrinkled yellow would be 3. And wrinkled green would be 1. We could write the phenotypic ratio as 9 to 3 to 3 to 1, meaning 9 round yellow, 3 wrinkled green, 3 wrinkled yellow, 1 wrinkled green. We could also write 9 out of 16, 3 out of 16, 3 out of 16, 1 out of 16. If you're wondering where the 16 comes from, there are 16 squares here. 1, 2, 3, 4. 1, 2, 3, 4. 4 times 4 is 16. So 4, 8, 12, and 16. That's going to be it. If you have any questions, please feel free to text me so I can clarify this. I am interested and expect you to make an A on this test. So please text me with any questions you have so you can earn that A. We'll see you guys in class.